You're going to send a different link again?
Yes, sir. Now it is time up, sir. This meeting is ended now, sir.
I'm going to say, have the animal type of characters, they have to protect themselves by some or other kind of mechanism. So one such mechanism we are going to discuss today in the form of plant secondary metabolites. So the major threat to the plants is from herbivores and many of the pathogens. Pathogen could be in the form of bacteria, virus, fungi, nematode, many of the insects or mites. And when we see the plants on the surface, they will be having a layer called cuticle, and following the cuticle, they have one more layer that is called periderm. So these two layers, one is cuticle, other one is a periderm. These two layers are very important in protecting the plant from enemies like bacteria and fungi. So in addition to this layers, that is cuticle and periderm, we have the special kind of organic compounds called secondary metabolites, which will help the plants defend themselves and to exist on the earth and combat with herbivores and pathogens by using a weapon called secondary metabolites. These secondary metabolites not only help in defend them from the various kind of enemies, they also give the support in the structure. As if we can see lignin, and we can see like anthocyanins. These two are the important type of secondary compounds which are not only playing role in defense but also in some of the structural role. So I am able to share you, I hope you are seeing the diagram where we can see the superficial layer. Just now what I spoke to you is about cuticle. This is, there are two bits in the figure you can see. A bit is a drawing and B bit is electron micrograph of a young leaf of Lamium, where we can see the cuticle layer, right? And the cuticle layer of a young leaf epidermis is shown there, where we can see that the different parts, whatever different parts are there, these different parts are shown to have an important defending compound called wax. As you might be knowing, wax is a kind of a lipid, and you might be knowing wax is a solid form of fat. It is a solid fat. You can see the simple fat, simple lipid, and a complex lipid. Your wax is a complex lipid with a different kind of alcohol. Lipid usually have two important things. One is alcohol, other one is a fatty acid. So if you see the more common simple lipid, you can see that triacylglycerol is a simple lipid, and when you see the wax is a complex kind of fat, and this is very important component in the cuticle, which is going to support or which is going to defend, which is going to help in defending the particularly plant from the various its enemies. So here the layers on the surface, you can see it's a cutin, suberin, and wax. Cutins are generally present on the above parts. Suberin is underground parts. Like we can see, plant has two parts. One is the aerial part, other one is the underground part. So here also we can see that this superficial layer, which is protecting the plants, is having again two types. One is underground, means I mean to say root kind of whatever root part is there. Root part is generally the underground part, and leaf, stem, and other parts are the aerial parts. So aerial parts are covered with a coating called cutin, and the root part or the underground part is having the suberin. And there are other parts also there in the plant which are having this tough layer of wax. So both the underground part and the the aerial part, both the parts are having the coating of wax. So this is one of the picture I am sharing. Here this picture is showing you the top layer of a cuticle where wax is clearly shown in scanning electron microscope, scanning electron micrograph, where we can see the different structures of the wax is there. The example is Brassica species, and the wax is shown in the form of here and there the crystal forms. And in one of the image you can see even some small tiny organisms are there. Hopefully they are some bacterial invaders which are present on the surface. The B of the figure, the B bit of the figure is showing you the invaders present in the form of bacteria. And now the plants are involved the plants are showing various interactions. Plants are showing various interactions. Like we can say, like pollination is one of the beautiful interactions where you can see the pollen transfer and where the fertilization process is beginning with the pollination. And here, secondary metabolites play role not only in defense but also it is going to play important role in the fertilization beginning process that is called pollination. So when did man has come to know about this story of defense, plant defense? So they told that when we check the fossil records, of the plants, it was at least 300 to 400 billion years ago when first vascular plant was evolved. At that time, there was also the evolution of the insects in the form of herbivores. So the story of Metabolites is a very old story. It is beginning in the Paleozoic period, where we can see the vascular plant as well as the arthropods. These arthropods are usually the one which are going to be the major herbivorous kind of animals. So now I am going to tell you something. What is a secondary metabolite? So when you see secondary metabolite, the name itself says second. We know first is primary and second is secondary. So we are going to divide the metabolites, the products, plant products into two halves. One of the half is primary. Primary means which has a direct role in growth and development are primary compounds or primary metabolites. And secondary metabolites are the one which has no role in the growth and development. Like there are so many growth and development related programs like photosynthesis, respiration, solute transport, etc. Whatever the list is given over there, these are the processes which are very important or essential for the growth and development. So secondary metabolites are generally different from the primary metabolites. Primary metabolites are usually amino acids, sugars, lipids, nucleotides, etc. But the secondary metabolites are not like amino acids, nucleotides, sugars, or lipids. They are different compounds, and they don't play any role in the growth and development. So here I am going to share you one more metabolism, how actually the primary metabolism is going to occur and how the secondary metabolism is going to occur. So here are the two colors given to you in the image. One of the image is showing you the pink color, other one is the blue color. The blue color says you the mechanism of secondary metabolism or secondary metabolite synthesis, and the above one you can see it is a primary metabolism. So here primary metabolism is routine processes where you can see your photosynthesis is involved, respiration is involved, your respiration has got glycolysis, your respiration has involved PCA cycle, your respiration has got involved with the HMP pathway, HMP shunt, and so many other mechanisms are there which is going to support. Primary metabolism is a pink portion, which is showing you the lot of processes which are going to help in the growth and the metabolism. And one which is showing you down. In the down, you can see the secondary metabolite synthesis processes. In the secondary metabolite synthesis process, here you are going that there are two major species existing in the biosphere. One is the plants, other is the insects. As you know, insects belong to the arthropods, and they are the dense or the largest among the living organisms in the whole biosystem, whole ecosystem. The whole ecosystem is densely populated. First, dense populated with the arthropods, and second dense population, second rank is for the plants. So plants are less compared to the arthropods or insects, but here the plants are vastly. They say that there are at least 4 lakh plus taxa or 3.5 lakh taxa. Whatever the number it is, the plants rank on the second position in the biosphere, and insects rank on the first positions. As the beginning of the session, I have been talking that these plants and the herbivores, that is arthropods, are closely associated. Both of them have got evolved in the Paleozoic period. And here the insects are again made in two categories. The insects related with plants, the insect having association with the plants can be made into two categories. One is either it is a pollinator or other it is a herbivore. Either they may be beneficial or they may be harmful. As you know, pollinators are useful and beneficial, and herbivores are, as usually you know, it is the enemy, or it is generally going to cause the damage to the plants. And these plants and the insects, whatever the insects which are either herbivores or pollinators, have evolved generally in the Paleozoic era, and in that era itself, plants have started their defense mechanism. They started fighting with this herbivores which are going to attack on the particularly plant's host. So plants generally have different type of strategies. They are having different strategies to protect themselves. Like just now I was talking to you about cuticle and periderm. So these cuticle and periderm is a mechanism by which the plants are trying to escape from the particularly herbivores or the insects which are going to cause the damage. And the other way of plants which is going to defend the insects or the one which cause damage is through biochemical traits. The biochemical traits itself, here we are going to talk, the biochemical traits itself is the particularly secondary metabolites. So these all mechanisms, these mechanisms could be generally consecutive or they may be adaptive. You may be knowing the genes are of two groups. Whatever genes are there, these genes are generally of two groups. One is the particularly group, one group is the particularly consecutive. So we are having adaptive responses and the particularly the consecutive responses. Consecutive responses are inbuilt. They are whether the enemy is present or whether enemy is not present. In both the situations, plants will synthesize the defensive compounds, but adaptive, generally we have adaptive system. Adaptive system will activate, or adaptive mechanism will activate during the enemy, whenever the predator or enemy attacks. So these are the two ways how the plants are going to respond: consecutive method, or it's an adaptive method. And there are number of, as I told you, there are number of morphological mechanisms to defend the particularly enemies like trichomes, spines, cuticles, thorns, especially trichomes and spines. I can talk about spines. Spines, you can see that many of the plants are having the particularly spines or thorns. Spines or thorns are generally making the enemy for any herbivore. For example, any herbivorous animal is going to feed the plant, then the thorns or spines will prick and the animal will be unable to unable to eat or feed that particular plant. So trichomes, spines, cuticles, thorns, lignified cell walls, all these are the first line defenses. These are the first line defenses which are helping the plant to protect or defend itself from the herbivores or the one which is going to feed the plant. And second one is able to increase its population. As you know, the insects are able to increase their population very fast. So a graying character or feeding character can be stopped. This mechanism, this method is called generally the anti-zoonosis character, and the growth, development or survival is affected. It is called the antibiosis. So usually the plants generally have the one important method, that is the volatiles. So you can see that if you see the flowers will release some decent perfume. So these one which is easy to spread is a volatile compound. A compound which is able to spread or is going to give the perfume is generally a volatile compound. Easy to spread is generally a volatile compound. So what the plants do is the plants release some kind of perfume, and these perfume will either stop the insect to come. If they will not attract, they will not make the insect to come nearby. Many of the plants, like you can see Miriam or you can see neem, they have got a kind of pungent kind of smell, and this smell itself makes the insect not to attack the plant. So generally, whatever the volatile is there, volatile sometimes attract and sometimes it will deter. Deter means keep away. So this whatever the defense is there, it is through the release of some chemical compounds. These chemical compounds are again by both methods. One is inducible method and consecutive method. I told you like a while before that inducible is whenever some enemy is available at the time, if at all the chemical compound is released, it is inducible. But if not, if the enemy is there, if the enemy is not there, in both the situations, if at all the defense is going to be there, that is consecutive. So like adaptive and consecutive, how consecutive is also called as housekeeping kind of character. So plant secondary metabolites are not required for normal growth, but the primary metabolites are playing important role for the particularly growth and development. And primary metabolites are precursors, means starting molecules. Secondary metabolites are made from the primary metabolites. Whatever primary metabolites, they are usually present as a housekeeping. They may be present whenever the herbivore comes and attacks. So secondary metabolites like the phytoanticipins and phytoalexins. Like I just now I told you a while before, I told you that there are two things: antibiosis and the anti-zoonosis. Antibiosis is usually the phytoanticipins, and anti-zoonosis is the phytoalexins. So there are a number of secondary metabolites in the plants, like in the pathway table and the pathway sheet I have shown you the primary metabolism is there and secondary metabolism is there. In the metabolism, I have shown you three important groups. One is terpenes, other one is the phenols, and the last one is the nitrogen-containing compounds. These all overall compounds, whatever three group of secondary metabolites are there, either terpenes, phenols, or the nitrogen-containing compounds, all put together, there are at least more than 1 lakh compounds are there, chemical structures. More than 1 lakh compounds are there in the different plant groups. Either it may be angiosperms or gymnosperms or the lower plants also even. So these secondary metabolites based on their structures could be alkaloids, amines, cyanogenic glucosides, glucosinolates, non-protein amino acids. I think you might be having the idea what are non-protein amino acids and protein amino acids. As you know, proteins are the polymers of amino acids, and the amino acids which are playing role in the synthesis of proteins are protein amino acids, and the amino acids which are having no role in the synthesis are non-protein amino acids. So alkaloids, amines, cyanogenic glucosides, glucosinolates, non-protein amino acids, organic acids, terpenes, phenols, ketones, polyacetylene, and peptides. These are the broad group of secondary metabolites which I have listed you. And as I earlier also told that there are at least 1 lakh chemical structures present in the secondary metabolite group. And generally they have told that these metabolites or these compounds not only play important role in the plant defense against the insects, but also they play important role in the stress, like abiotic stress. Different kind of abiotic stresses are there. In this abiotic stress also, these chemical structures are going to help the plant or the survival of the plant. They are very important. And these to adapt in particular ecosystem or resist from particular insect pests, but through their chemical defenses. As I told you in the earlier slide also, these secondary metabolites play important role even to attract the insects. I have made insects into two categories in the early part, that is herbivores and pollinators. So herbivores are the one which is harmful, and pollinators are one which are useful or beneficial. So pollinators are also getting attracted with the same things. The secondary metabolites have got dual role. One is attraction, other one is the deterrence. So pollinator insects are going to attract it with the smell and color of the flowers. So these colors and the smell, both these are coming under the secondary metabolites, which are helping the plant to attract the insects and get done their fertilization process. So, as you know that number of plants are there in the flowering plants. We have flowering plants and non-flowering plants. If we see the flowering plants are again getting blown in the night time, in the daytime. If you are seeing the flowers in the night time, generally the nighttime flowers are white in color and daytime flowers are colored. And daytime flowers are multicolored and they will attract the insects by their colors. But nighttime flowers are white and they will attract the insects through their secretions, which are having a particular kind of secretion called nectar. Nectar is a secretion which is having sweet in taste, sugar. It is sugar-rich compound which is attracting the insect pollinator towards them. So these flowers are having color or having nectar. So these both nectar and the particularly color, both is due to the secondary metabolites. What are secondary metabolites are they? Secondary metabolites are playing important role in attracting the pollinator kind of insects. And they have told that these insects, approximately there are 80% of the herbivores are there which attack a particular plant species or a particular single genus or a family. Like you can see, some plants, some insects attack groundnut, some insects attack paddy, some insects attack some kind of. So these insects, when we talk about, as I told you, the herbivores, When I'm talking about herbivores, 80% of the herbivores are there which attack a single group of plants. They will not go into the different plants. For example, like midges. Recently, we have heard the story of midges in Gujarat or some other places from different countries. Midges come attack, and slowly they have moved into Maharashtra to our state borders also they have reached. So these midges are particular kind of herbivores. We can talk them as to be, we can consider as the recent example of herbivores, and they are particularly attacking a group of plants, single genus. So among the herbivores, 80% of the herbivores are there. They are known or they are specialized to attack a particular group of plants, a particular family or a particular genus of plant. So like I will be moving a little bit rushing. So here we are seeing that terpenes are there. As I shown you in the tape in the flow sheet, there are three groups of secondary metabolites: terpenes, phenols, and nitrogen compounds. Terpenes are the largest group of, among the three kind of, among the three type of three major group of secondary metabolites. Terpene are the largest group, and they are made from the glycolytic pathway, that is the acetyl-CoA precursor, and phenols are there. We have about 10,000 phenolic compounds are there, which are not soluble in water, and terpenes are also non-soluble in water. And instead of solubilizing in water, they are non-polar. They'll be dissolving in the organic solvents. And then comes is nitrogen compounds. Among the nitrogen compounds, as I told you, the nitrogen compounds are made again into two groups. One is the alkaloids, other one is the cyanogenic glycosides. It is very interesting now to see. What is very interesting is cyanogenic glycosides. What are these cyanogenic glycosides? First, I'll talk to you a brief 2 lines about the alkaloids. Alkaloids are the nitrogen compounds. They are organic nitrogen compounds, and there are at least 1,500 metabolites which come under the category of alkaloids, and 20% of the species of vascular plants are known to contain the alkaloids, and these alkaloids are usually having heterocyclic ring. Heterocyclic ring, you might know, one benzene ring and one pyrrole ring. So this is a heterocyclic ring which is primary structure present in the alkaloids. So these are some of the alkaloids, major alkaloids which I am sharing with you, like pyrrolidine, propane, piperidine, pyrrole is dine, quinol is dine, isoquinolone, indole. So these are the major group of 7 different alkaloids, and their examples are given. As you know, nicotine. Nicotine is a pyrrolidine, and ornithine, it is a propane. Like the cocaine, propane is a cocaine, atropine. So there are different kind of alkaloids given, major alkaloids given in the particular table. And what is cyanogenic type of glycosides? What do you mean by cyanogenic glycosides? So here the cyanogenic glycosides are made into 2 types. One is cyanogenic glycosides, other one is the glucosinolates. What are these? These are poisons. Why they are poisons? Because they are going to release some toxic compound or toxic gas that is called hydrogen cyanide. So hydrogen cyanide releasing nitrogen compounds are cyanogenic glycosides. They are going to release the hydrogen cyanide, and they will be killing the enemies, or they will be killing the herbivores. And the second group is there, that is called glucosinolates. These glucosinolates are not going to release any kind of hydrogen cyanide. Instead of cyanide, they are going to either give some smell or they will give some typical taste. These typical taste or typical smell will make the insect to keep away. The insect is kept away from the particular plant. So usually they told that glucosinolate is having the smell of mustard oil. So you are having a lot of vegetables like cabbage, broccoli, radish, etc. These are the Brassicaceae members which are well known to give the glucosinolates and off so that the insects can't attack them, herbivores can't attack them. So this is a list in a graph shown where we can see the plant secondary metabolites of different categories. You can see the abundant among the bar. You can see the blue bars on the screen. So in this blue bars, the largest bar in the bottom is alkaloids, and the smallest bar you can see the amines, cyanogenic glycosides, glucosinolates, and carbohydrates. There are 4 different kind of primary secondary metabolites which are less compared to alkaloids and terpenoids. Alkaloids and terpenoids are the less number of secondary metabolites present in the plant kingdom. The rest is first rank alkaloids, second terpenoids, third is polyphenols, peptides, polyketides, polyenes. These are the different other group of secondary metabolites which are given in the graph. And you can see the y-axis is the name and the x-axis is the number. X-axis is the number of secondary metabolites given, and the y-axis given the names or types of the secondary metabolites. So here is the sheet where the role of this particularly secondary metabolites is given. You can see the different colors. In these different colors, I am able to show you the top ones are the defense mechanisms and the bottom ones are the attracting role. One is the deterrency, above one is the deterrency, and the below one is the attraction towards the plants, and keeping away from the plants is the upper one, and attracting towards the plant is the down one. So here is the overall picture where you can see how the secondary metabolites are playing important role in the various mechanisms. So you can see the various mechanisms like the pollination and how the ants, especially some ants, are they going to attack the plant species. So these secondary metabolites will also help particularly better the defense the particularly ants, and you can see even they are going to play important role. I mean, plants themselves have some important plant. I mean, it's a group of plants called insectivorous plants which will trap the plants, and there are some in the top. You can see in the top you can see the insects are going to infect the plants and cause the infestations in the form of galls or infect the fruit. You might be knowing very good example of fig. Fig is a very good example of the particularly interaction between the beneficial insect and the particularly the fruit formation. And one of the major cuscuta, the yellow in the top, last one you can see cuscuta. It is a parasite. Okay, so here is the entire picture, and bottom ones you can see the association between the different I want to say symbiosis. You can see nodule, bacteria, and the root lichen, association between the fungi and the algae. Then again you can see coralline root association where you can see the roots, aerial roots, and the association with some cyanobacteria. So this is the overall method how the secondary metabolites are playing role in parasitism, how they are playing role in antagonism, how they are showing role in symbiosis, mutualism. In all these plant interactions, secondary metabolites are playing key role. This picture is showing the role of secondary metabolites in various interactions of the plants with the ecosystem. So these are some important kind of compounds found in the plant species. Like first one is a defense, and I'm able to show you the compounds and the examples in the particular plant species. And then they are going to play important role in the particular signaling molecules for the parasite. Parasite will come to know about the host, but through this, like striga actones, very good example. And then comes this orobancol, which is going to orobank, and other example is also given to you. And then not only this, secondary metabolites are also having the antimicrobial role, and the name antimicrobial and antifungal role, where you can see the things, and even in pollination also they are going to play important role. So the pollinating plants will release secondary metabolites even there, like caffeine, then gallic acid, nicotine, etc. In the different plant species they are going to make. And the most important thing is poison and deterrency. You can see the pathogens group, or we can see that we are going to grow these plants in our say surroundings or in the lawn area, so that most of the insects should not enter the home. You can, if you are growing this plants like pathogens group, chrysanthemum group in your lawn, automatically the pathogens is having a kind of typical secondary metabolite which is avoiding the entry of your insects or mosquitoes in the home. So like this, the beneficial role of Tagetes. And I am going to show you one table. In this table, I am going to show you some important examples of the Indian plants and from where they have originated and what are the effects they are going to show. One of the important member, Ageratum species from Asteraceae, which is highly allelopathic, and Argemone, it is harming the flora through allelopathy, and then Ecornia is there, and then Lantana is there, Parthenium is there, Prosopis is there. So these are the plants which are native from different countries, and they are having strong allelochemical effect, allelopathic effect on the crop plants. I mean to say, these plants will not allow allelopathy. You might be knowing allelopathy. Like if you are suppose seeing, we may be friends with some of them, we may be not good, or we may not be friends with them. Same happens in case of plants also. Plants may have some good association with other plants, and sometimes they may not have good association. If they don't have the good association, what they do is they release some chemical compound, and these chemical compound will prevent the growth or germination of the plant. This mechanism of avoiding their other plants, neighboring plants, is allelopathy. Allelopathy is a mechanism by which the plants will not allow to grow the other plants. You will be knowing very good that if you go under the banyan tree will not allow to grow the other plants. Like banyan tree or any of the shady trees, you see under the shady trees, trees will not allow the growth of any of the plants under their shade, because they are able to release some chemical compound, and this chemical compound will avoid the germination. Such mechanism of plants to avoid the growth of other plants is called allelopathy. So this is the best example. I'm showing you in the table 6 examples which are very well commonly known in the India, like Ageratum, Argemone, Ecornia, Lantana, Parthenium, and Prosopis. These are the well-known allelopathic plants which will affect the growth of our crop plants. These plants are not from our Indian origin, but they are from the other origin. But these are the very popular plants growing nearby to our surrounding crop. Wherever you are putting a paddy crop, or when you are putting a maize crop, or any of the crop, when you are growing the crop, these kind of plants are very familiar, and they will be avoiding your growth of the crop plants. So this is an overall picture which is showing you the role of secondary metabolites, where they can avoid the insects, and they may, they may be inducing the insects. Sometimes they may be deterring the insects. So this picture, whatever this picture is said, this picture is showing you how the secondary metabolites are going to counterattack the insects through their various mechanisms. Like they may be playing, the secondary metabolites may be playing the role as enzyme inhibitors. They may be blocking the membranes They may be sometimes causing the toxicity. They may be causing sometimes cell disruption. Likewise, these are the roles, various roles, how the secondary metabolites or plants are going to avoid the particularly insects. And there is some other role is also there where these are going to support the insects. And now this is one of the picture where it is shown. Initially, the larva of one particular insect has attacked the plant tissues, and you can see in the picture the larva is biting the stem, and immediately after the biting of the stem, the plant is releasing some latex. You know latex, milky secretion, specialized kind of fluid tissue it is. Latex secretion is released from the plants, and that is causing the death by poisoning or by miring the insect. So these are the pictures of some of the pictures of the insects. These are some of the insects which are going to the harmful insects, herbivores, which are going to attack the plants. These pictures, these insects are from different countries like Czech Republic, Japan, again from Czech Republic, then again from New Guinea, Japan, like this from different countries. These are the particularly insects which are going to attack the plants. And next picture you can see that these insects are going to attack the leaf. I think you might be knowing very well good example of these galls. These galls are very common in our surroundings also, Pongania. You can see the Pongania. In Pongania also you can see that lot of insects are going to attack the Pongania, and this Pongania is going to get the galls. Similar kind of one example I am showing you in this image, where the galls are made by the herbivores, which are the plants which are failing to release the secondary metabolites are going to get affected in this way by this feeding insects. And this is my last slide. And from this last slide, I am going to show you these insects are not only going to cause the harm, but in one of the beautiful example, there is a female insect which is shown you in the image A. In the image A, you are able to see a female insect that is Chrysomela species, and these Chrysomela species is going to particularly lay its eggs on the particularly twig of a plant called poplar plant. And this poplar plant is going to lay the eggs and it's a leaf base in the lower epidermis of the leaf. Female insect lays the eggs, and now we can see that larvae are going to emerge from the eggs. And whatever these larvae are there, these larvae are going to initiate, whatever the larvae are there, larvae are going to initiate lot of salicylic acid in the leaf. So when salicylic acid is increased, the salicylic acid is increased, the salicylic acid converts into salicyl aldehyde that is released from the abdomen of this larva, and these salicylic compounds are going to protect the plant from the invertebrate predators. What are the predators in future going to attack the plant are going to be get protected. So this Chrysomela is going to be a beneficial insect which is coming on the plant and protecting the plant.
Excuse me, sir. Sir, please.
Yes.
Sir,
yeah, I will wind
this session is enough, or we need another session? Only 3 minutes, sir. 3 minutes are left, sir.
Okay, I'll finish. I will wind
okay,
up.
sir. Thank you, sir.
So here with this, the major aim of my today's presentation was to tell you my research work. That is, I'm working on the secondary metabolites. I'm synthesizing the secondary metabolites from the particularly endophytic fungi. What are the endophytic fungi? Fungi which are going to inhabit the plant, leaf, stem, root, etc. And by living inside the plant system, these endophytic fungi will acquire the ability to produce the secondary metabolites. So in future, I may be sharing my work on secondary metabolite production through the particularly endophytic fungi. With this, I come to an end of my presentation. I express my heartfelt thanks to Tirupati sir and principal of that particular college, GDC Women Jagitial, and the entire department of Botany, GDC Women Jagitial, and Carsten Group behind this webinar, and the entire participant. I express my deep sense of gratitude to all the people who have been very patient to be with me during my presentation. Thank you so much.
Okay, sir. Thank you very much, sir, for giving such a wonderful presentation on secondary metabolites. This is the very most important topic, physiology, especially in plant physiology. Secondary metabolites are very important and very prominent role in all physiological functions related to pollination and different activities. We are very happy and more enlightened. We hope all the participants are enjoy your session, sir, and very beautiful images and beautiful slides in front of us, sir. We are and our participants are requested to please share these PPTs for our participants. We hope you sent this PPT, sir, and thank you very much. And now, before I conclude, our college staff club secretary and task coordinator, M. Satyaprakash sir garu, please give a brief vote of thanks, sir. Please welcome.
Okay. Thank you, Tirupati. And behalf of GDC Women, we pay sincere thanks to the Dr. Ahmed Abdul Haleemkhan sir. Sir, by your lecture, the session made very sound, and the entire faculty and the participants are enriched by your lecture. For this lecture, we pay our sincere thanks to one and all, and moreover, I congratulate all the participants for their participation in this webinar. So thank you, sir. Thank you very much.
Okay, sir. Thank you very much, the M. Satyaprakash sir. And now I would like to thanks all the family of Government Degree College for Women for success of this webinar, and also we are wholeheartedly thank you very much today's eminent professors, speakers, Professor B. Vidyavardhani Madam Garu and Ahmed Abdul Haleemkhan sir Garu, Head Department of Botany, Telangana University, Nizamabad, our entire GDC Women Jagitial, and all participants. We are very thankful to you, sir. And also, I am very thankful to our college principal, M. Srinivas Reddy sir, Vice Principal, Masru Sultana Madam Garu, IKC Coordinator, Dr. K. Kiranmay Madam Garu, Academic Coordinator, A. Shankaraya sir Garu, NSS Coordinator, Dr. M. Madhukar Rao sir Garu, and A. Rajani Madam Garu, Librarian, Mana TV, Rapat Srinivas sir Garu, and Staff Club Secretary and Task Coordinator, M. Satyapati, and our Women Empowerment Cell, T. Swaruparani Madam Garu, Zoology Department, Dr. T. Mahesh sir Garu, Chemistry, Dr. T. Karnakar sir, G. Manasa and GVR Sai Madhukar sir Garu, and Computer Science, Satyam, B. Sangeeta, Manasa, and Swapna Madam Garu, and English Department, D. Sunita, and E. Josna Madam Garu, and non-teaching of our college. Along with this, my Professor Dr. T. Christopher Ruben, Kakati University, Warangal, and my research lab mates, G. Rajender and K. Srikanth, D. Sujata Madam Garu, Assistant Professor of Botany, Government Degree College for Women, Karimnagar, D. Sujata Madam Garu, and Deepika, and Dr. K. Rajender sir, and Dr. B. Prasad sir, and Dr. Chitakari Ravi, and other lab mates. And my sincere thanks to my brother, beloved brother, Devaraju Srinivas, assistant professor and academic consultant in Telangana University Department of Botany, and my beloved brothers and my brother's classmate, my brother Vemula Suresh and T.N. Swami. They are working as assistant professor, Giriraj Government Degree College, Government PG College, Nizamabad. And my sincere thanks to all the participants, your patience, and I'm very extremely sorry to say, because of this interrupted webinar, but you are patiently participated more than 3 times, and we are wholeheartedly thankful to you. And lastly, but not least, my department, Srimati B. Sandhya Rani Madam Garu, and economics department, V. Jamuna Madam Garu, and history, Madhavi Madam Garu, and my entire GDC family. I'm very, on behalf of Department of Botany, wholeheartedly thankful to everyone, each and every staff member and non-teaching staff members of our Government Degree College for Women, Jagityal, and also today's chief guest, Dr. Balabhaskar Sir Garu, AGO CCE Hyderabad, and our today's guest of honor, Dr. K. Surendra Reddy Sir Garu, General Secretary, TGCGTA, Telangana State. On behalf of Government Degree College for Women, wholeheartedly thankful to you, sir, because of your encouragement and motivation. We are unforgettable, sir. And all the dignitaries and different college lecturers and professors and principals of degree colleges, on behalf of Government Degree College for Women, wholeheartedly thankful to you, sir, and thankful to you, madams. I'm already feedback link is sent in our chart, madam, and also feedback link and this PPTs, this webinar PPTs, we will send in our webinar group, and also sent if sir and madam will send to me automatically, then I will send to you, sir, definitely. And this video is uploading most probably today or tomorrow in YouTube also. This is for because of our subject enrichment, especially in degree and PG colleges. That's why I am very happy and thankful to you, each and every participant of different states of our India, and all colleges and entire Department of Botany fraternity faculty, wholeheartedly thankful to you, each and everyone. Thank you very much. Okay, I will conclude. I will end the session because of time is up. Within 10 seconds, it will going to end, madam. Thank you very much.